Hi, I'm Riccardo, and this is our work regarding rat sensitivity to multipoint correlations. Natural images contain complex structures and regularities that make them really different from randomly generated ones. In particular, we focus on the complex correlations in the local distribution of light across pixels, as these are fundamental for the perception of edges and features that are present in an image. This is clear from the example presented here, where, despite removing all low-level features, it's still possible to recognize the original subject of the image. Now, if we reduce our analysis to groups of up to 4 pixels and we scan the image through a 2x2 window, we can measure the frequency of 16 possible combinations of black and white pixels. We can then reduce these 16 combinations to 10 unique and independent configurations. Measuring the variability of these correlations across many patches of natural images revealed the presence of a specific ranking, with 2-point correlations being more variable than 4-point correlations that are more variable than 3-point correlations. This ranking is particularly interesting because it's a unique property of natural images and is not found in random noise. So if we now look at sensory perception, according to the efficient coding principle, we expect that the visual cortex should allocate more resources to encode features that are more variable and therefore more informative. Following this idea, different behavioral studies on humans have been conducted to measure sensitivity to the multipoint correlations seen previously. All used the stimuli maximum entropy textures, as the one shown here. These are artificial textures where the value of one of the correlations is decided, and all the others are implicitly fixed by the condition that the texture has to have maximum entropy. These textures are convenient to measure the sensitivity to the correlations in exam, as they don't contain any undetectable correlation between the statistics. The results obtained from these human studies revealed that the sensitivity to multipoint correlations was recorded exactly as the variability in natural images. The results of the human studies seem to indicate that the human visual system is adapted to the environment as expected in the efficient coding framework. But in humans, it's not possible to perform any direct causal study of this adaptation, as it would be possible to do with rodents through controlled rearing experiments. Still, going to the main goal of this work, it's not known if rats share with humans the same sensitivity to multipoint correlations. To answer this question, we selected 4 image statistics from 1 to 4 points and used the stimuli maximum entropy textures as shown here with their building blocks. For each of the statistics, we trained a group of animals in a discrimination task where they had to distinguish between textures containing the correlation and white noise pattern. Animals autonomously triggered the presentation of a stimulus and then had to click left or right, depending if they judged the texture as containing the correlation or only noise. Here, we have as an example the results obtained from one of the rats trained on 2-point correlations. As you can see, increasing the intensity of the statistic leads to an increase in the fraction of times the animal judged the texture as containing the statistic itself. To infer rat sensitivity from this data, we developed an ideal observer model that would yield a psychometric curve for each rat. This model only depends on 2 parameters that are unique for each animal, and are the animal prior bias and the measure of the perceptual sensitivity, loosely related to the slope of the final curve. As you can see for this example rat, the model yielded a sigmoidal curve that fitted very well the data points. When we fitted with our model the data from all the rats, we obtained psychometric curves that differed quite a lot in terms of steepness of the slope, depending on the multipoint statistics the animal were trained to discriminate. If we now plot the perceptual sensitivities that can be derived from these psychometrics, we can see that their distributions differ substantially across the 4 groups. And if we look at the ranking, we find that it is the same already observed for humans and in the variability of multipoint correlations in natural images. And for the set of statistics that were studied both here and in the previous studies, the actual values of the rat sensitivity matched up to a scaling factor, both the human sensitivity and the standard deviation of the statistics in natural images. To further validate these findings, we performed an additional within group and within subject comparisons. To this end, each group of animals was either tested with a new statistic or was split into 2 subgroups, each tested with a different statistics. Results of this analysis are reported in the figure presented here. Colored points with halo show the average sensitivities of the groups of rats to the statistics they were originally trained on. The other colored symbols connected by a line show the average sensitivities obtained when subgroups of rats originally trained on a given statistic were tested with a different statistic. Sensitivities achieved by individual animals are instead presented as shaded data points with the corresponding symbol color combination. rats trained on 1 and 2 point statistics performed poorly with higher order correlations, while animals trained on the 4 point statistic performed on 2 point correlations, as well as the rats that were originally trained on those textures. These show that the better discriminability of textures containing lower order correlations is a robust phenomenon, which is independent of the history of training and observable within individual subjects. Moreover, performance on 4 point correlations was higher than performance on 3 point correlations for each group of rats. This was true in particular not only for rats trained on 4 point and switching to 3 point correlations, but even for rats trained on 3 point and switching to 4 point, as you can see comparing the green versus the purple triangle. This means that the larger discriminability of the 4 point statistic as compared to the 3.1 is again a robust phenomenon within individual subjects. These results provide the first demonstration that rats and humans are similarly adapted to process the statistical structure of visual textures, confirming the importance of these multipoint correlations in shaping visual perception.